Let's look into molecular phylogenetics, where we look into the relationship between the species and the actual molecular units from which this evolution passes through, or from which this evolution is carried into the next generations, that is the genes. So the actual pattern of evolutionary history is the phylogeny or evolutionary tree with which we try to estimate. So a tree is a mathematical structure which is used to model the actual history of a group of sequences or organisms. So there are three different forces which work in modifying this information which is present in the genome, or they try to change the genes. One of them is these expansions, that is the genes they get duplicated. The other one is deletions where the genes are lost. And the third one is horizontal gene transfer, or HGT, where the genes are transferred between different species. So for example, sometimes different microorganisms they shift the genes between different species, or they can carry the genes of one species. So in this way we can have these horizontal gene transfers. So molecular phylogenetical methods they allow the construction of phylogenetic trees from a given set of aligned sequences, while aiming at reconstructing the history of successive divergence which took place during the evolution between the considered sequences and their common ancestors. These trees may not be the same as the true trees, but in this way you can have a predictions, or you can estimate the actual trees while using these evolutionary trees which you reconstruct. So let's look into this situation. So we start with an original gene, and then in the final species we have species 1 where we have A1 B1, and in second species we have A2 B2. When we make their trees, we feel like the B1 in one species goes with the B2 in the second species. Same way A1 and A2 they are clustered together, and we believe they have a common ancestor A, whereas B2 and B1 might have somewhere they have B, and before that we take both these A and B and they were actually coming from some original gene O. So if we look into A1 B1 A2 B2, all these things are homologs with one another. If we talk about A1 versus B1, since they are similar genes and they can be the diverged and they are present within the same species, we can call them as paralogs. So these two they are paralogs, and these two they are paralogs. Whereas A1 and A2 are orthologs, they are present in different species but they are similar copies. So this A1 and this A2 they are orthologs. Same way B1 and B2 they are orthologs. So when we put these genes together and when we construct their evolutionary tree, we can go back and trace their evolutionary history. In this slide we see that the genes they get duplicated, and then these duplicated copies they can further diverge from one another. So this gene duplication as well as divergence can lead to speciation, so it can lead to formation of new species. So in this way, when we put those genes together, the actual genes, we put them together in a tree, we can trace back the history of speciation, and we can move back and where we can find where we might have expected some gene duplications over the period of time. Reconstruction of a phylogenetic tree is a statistical problem, and a reconstructed tree is an estimate of the true tree. So it is important to get an idea or do some statistical testing to find whether our estimate is correct or it is not correct. If we look into nucleotides versus amino acids, so which tree is better? So the idea is which one is more informative, where we can expect more divergences, where we can expect more patterns, or we can have more information where we can find the differences between two species, that is the good one. So in this case, if we see the nucleotide sequences, they have higher information content than amino acids. How? let's look into it with this example. So we see that there are some synonymous changes. Synonymous changes are where we can have an amino acids which are coming from different codons which are present in the DNA. So we might have differences between the nucleotides, but since more amino acids are sharing different those nucleotide codons, the amino acids they remain the same. So if we are correlating two amino acids with one another, we might not find the differences, but if you go back and compare their nucleotides, you might find the differences. So in this way, these DNAs, if you make the DNA based trees, you can get more phylogenetic informations than if you get the tree from proteins or amino acids. Here is the synonymous changes versus non-synonymous mutations. Synonymous changes, they alter the DNA sequence, but they do not affect the amino acid sequences. So for example, GGA, and in the second strand you have GGA. So you cannot, here we don't have any information since glycine and glycine they are matching. In the second you have GGC, where you have GCA, but if you compare their amino acids, you would say that they are the same. So in this way your alignment is good, and these are species where you believe or these sequences are coming from the organisms which you believe they are same or similar, since you cannot find the differences. But if you go into the DNA level, you will see there is a mismatch. The third nucleotide was C, and here the third nucleotide was A. Same way, ATA gives you isoleucine, whereas ATT also gives you isoleucine. So the concept which we are trying to elaborate over here is that if you make the tree from DNA, that is more informative. So molecular phylogenetics is the study of evolutionary relationships between genes and species.